And I wanted to thank USGS again for webcasting and for making these video files available for folks who couldn't travel, many folks from Oregon and Washington who couldn't travel to this meeting this week. I know we had some computer problems yesterday, so our first speaker, Richard Feely, brought his Macintosh, so we'll see if that works any better. So, but this morning we will be focused on specific effects to species, and Richard Feely will start that off on the issue of ocean acidification and its effects to species and ecosystems. Richard is a supervisory oceanographer for NOAA at the Pacific Marine Environmental Laboratory. His research interests include chemical oceanography and aquatic chemistry, specifically the mechanisms controlling sources and sinks of man-made CO2 in the oceans and the impacts of hydrothermal processes on the chemistry of the oceans. Richard is responsible for the development of chemical oceanographic research studies for several of NOAA's national research programs, and he's also responsible for directing the development of analytical procedures for the analysis of CO2 in seawater. Richard is a widely published author, and he sits on 9 national and international committees and panels, and I won't list them here, on oceanography and climate change. And he holds many awards, including being co-awarded for the 2007 Nobel Peace Prize with other IPCC authors. So please join me in welcoming Richard. Come on up.
Well, thank you very much for that wonderful introduction, David, and thank the U.S. Fish and Wildlife Service and the U.S. Geological Survey for inviting me to be here. I'm the other kid in the block talking about the other CO2 problem, that is ocean acidification. And when I start these talks, I always first present this first slide. Most of you know automatically what it is. It's probably the most well-known slide in the world on the carbon system. It's the Mauna Loa record from Dave Keeling, who started his work in 1957 and continued it out through 2007, showing the increase in atmospheric CO2. And what I've superimposed on that slide is the corresponding Hawaiian Ocean time series site at the Aloha site just north of Oahu. And the dark blue color is the PCO2, the concentration of CO2 in surface seawater, and the cyan color is the pH. And for measurements like this throughout the world, we know for certain that the CO2 in the oceans is increasing at a rate that's commensurate with the atmospheric increase, and the pH is decreasing on the order of 0.02 pH units per decade. So we have data throughout the world at many, many stations that allow us to ascertain that this is a very consistent pattern throughout the world. And we know quite well that the ocean uptake of this anthropogenic CO2. So what I will talk about today is what is ocean acidification, what is the extent of ocean acidification, and some of the impacts on our marine ecosystems. Now, if we look at the IPCC models and their predictions of what has been happening, we know that from land use changes, we have released about 160 billion metric tons of carbon into the atmosphere. And in addition to that, through fossil CO2 burning, we released another 348 billion metric tons of carbon into the atmosphere. And most of that resides in the atmosphere, about 234 billion metric tons, or about 46%. The terrestrial sink is about 147 petagrams of carbon, and the oceanic uptake is about 127. This is through the year 2000. And we have thought over the last several decades that the ocean uptake of CO2 was a good thing because it reduces the CO2 concentration in the atmosphere by about 70 parts per million and cause an overall cooling of the atmosphere by about half a degree C or so. But what we're now finding that this uptake of this amount of carbon in the ocean is having a very serious and deleterious impact on our ocean ecosystems. Here is an example of what we have for our atmospheric network throughout the world. This is run by NOAA's Earth Research Systems Laboratory. And we can see from the number of measurements that we have, starting from the South Pole all the way to the North Pole, we can actually see the breathing of the world, the uptake of CO2 in the summertime and its release due to respiration in the wintertime. But you can see just from this small 10-year record, the gradual increase in the world baseline for CO2, increasing now at a rate which is unprecedented of 2 parts per million per year. If you take the IPC scenarios for CO2 emissions out through the rest of this century, and I've shown here a number of different emission scenarios, you can see that if we want to sustain CO2 at about 450 parts per million in the atmosphere, we need to begin to reduce CO2 emissions right away, very dramatically. If we want to sustain CO2 emissions at 550 parts per million, we need to begin with the next few years to begin to reduce CO2 emissions. Whatever we decide is the sustainable level of CO2 in the atmosphere, we should note that the change in the CO2 in the oceans will be with us for many hundreds of years to thousands of years. It's because of the slow turnaround in the oceans and the very slow change in the chemistry that takes place. And for pH, for example, that means that we are developing a new baseline of what the background pH levels that we will have to deal with for the next several centuries. And this ranges anywhere from 0.1 to 0.5 pH changes that we will be dealing with, depending on what sustainable level of CO2 we choose to approach. So how does CO2 ocean acidification work? CO2 reacts with seawater. It exchanges across the air-sea interface and reacts with water. That reaction is very, very fast. It forms carbonic acid. Carbonic is a weak acid which immediately dissociates to form a hydrogen ion. That hydrogen ion is what gives us our acidity. That hydrogen ion immediately reacts with a carbonate ion in seawater, consuming the carbonate and producing bicarbonate. It's the carbonate species that is very important for the formation of all the skeletons and shells of our carbonate-forming organisms in sea. And you can see here an example. The white cliffs of Dover on the bottom picture is just full of coccolithophorids, which are plant materials that form these calcium carbonate shells. So the base of this food chain is directly affected. If we look over time, we see that the pH of seawater has dropped on average from 8.2 to 8.1 since the beginning of the Industrial Revolution. Carbonate ion concentration has already dropped 16%. If we project the IPC scenarios out to the end of the century, we would see a pH drop of another 0.3 or 0.4 pH units, down to about 7.8. This would be a 150 to 200% increase in the acidity of the oceans because of the addition of anthropogenic CO2. And the carbonate ion concentration would drop to about 50% of what it is per day. So you can see that the organisms that produce their calcium carbonate shells will have a more difficult time to do so. Now, the organisms themselves are concerned about the saturation state of seawater. And in order for them to produce their shells, they have to have a high saturation state, generally around 6 for calcite or 4 for aragonite. And this is very important for them to have a high saturation state to produce their shells. If the saturation state is a function of the calcium concentration times the carbonate concentration divided by this apparent solubility product. This apparent solubility product increases with decreasing temperature and increasing pressure, so that in the cold waters of the deep ocean, the apparent solubility product is high and the saturation state is low. This is why we have dissolution of calcium carbonate in the deep ocean. In the surface warm waters, the saturation state is high, and as I said, generally between 4 and 6. Now, as we approach a equilibrium value of 1, what we see is slow down as a calcification process, and below 1, a dissolution begins to occur even while the organism is still alive in some cases. Now, we have conducted a complete survey of the carbon system in the ocean in the 1990s in order to determine the amount of anthropogenic CO2 in the ocean. This was 99 cruises conducted by 8 different countries. We brought together all the data in my laboratory, we corrected all the data, and provided a unique data set with a high degree of precision and accuracy, over 72,000 samples. It took us 5 years to develop all this data set. And what we found is that we could determine the distribution of anthropogenic CO2 in the oceans, and you can see here from the Indian and Pacific profiles that most of the anthropogenic CO2 resides in the upper 1,500 meters of the water column. With the exception of the North Atlantic, where it penetrates all the way to the bottom, we still have the condition where anthropogenic CO2 still resides in the upper part of the water column, where most of our organisms of concern still live. And this anthropogenic CO2 is causing a shoaling of this aragonite and calcite saturation horizons towards the surface. So where this corrosive water is approaching, we are already seeing the effects of anthropogenic CO2 on the corrosiveness of the water towards the surface. And here I show the migration of those surfaces towards the surface in the Indian and the Pacific, with the very shallow regions of the North Pacific here showing very shallow corrosive waters as close as 100 meters in the North Pacific by Alaska. So if we map the distribution of this corrosive water, we can see the global impacts. And here we see that it's still deep in the North Atlantic, about 2,500 meters for aragonite, 3,500 meters for calcite, except for this region, the eastern North Atlantic. And in the Pacific, you can see it's quite shallow. As I said, it's very shallow around Alaska, but along our coast of Washington, Oregon, and California, and in South America, it's abutting right against the continental shelf. So any changes to the circulation and upwelling of water onto the surface will cause this corrosive water to come onto our continental shelf. So what we did was we worked with Jim Orr and developed a map of the distribution of this aragonite saturation for the world oceans. The blue colors are very safe colors for organisms. The cyan or magenta are conditions where calcifying organisms, particularly coral reef systems, will begin to have problems, and when you get in the yellow and reds, they're in severe stress. What we find with the model projecting from 1775 is that out to 2100, with a business as usual scenario, the entire world oceans will be under stress for ocean acidification, and the high Arctic regions and the high Antarctic regions will be in undersaturated waters by the end of this century. The Arctic Ocean was not included in this, but in fact the Arctic Ocean will be the first to see corrosiveness, and by 2050 the entire Arctic Oceans will be in undersaturated waters, fastest of all the ocean basins. So we have to concern ourselves with some of the natural processes that can accelerate this process. And along our coast, there's a natural coastal upwelling process that occurs in the summertime. It's diagrammed here. When the winds come from the northwest, the surface waters are pushed offshore and the deeper waters are moved onto shore. And this upwelling process occurs from Canada to Mexico. And so in 2007, we were able to have a cruise to define how this impacts our continental shelf along our coast. And what we found was, indeed, we do have an upwelling process. This first slide shows the temperature and density. And on these density surfaces, we see very cold waters less than 9 degrees being pushed onto our shelf, and in some place moving all the way to the surface and all the way to the coast. These waters carry with it the corrosive undersaturated waters, the waters with the saturation data less than 1.0. So these corrosive waters are on our continental shelf right now, and the pH of those waters are less than 7.7. What we would expect for models to occur at the end of the century is occurring right now on our continental shelf in our coastal region. And so we mapped this out at the surface in 120 meters. And you see at 120 meters, this corrosive water is everywhere we looked Canada to Mexico and probably proceeds all the way down to South America. And we can see evidence of this in the surface waters in the blue areas you see there upwelling on our continental shelf. We have had colleagues, Nikki Gruber from UCLA and colleagues, model this, and you can see on the right-hand panel the corrosive low pH water in blue upwelling during the summer months all the way along our entire coast and then vecting out into the surface waters to the west. So this is an upwelling process that occurs anywhere from March through November and occurs all the way along our entire continental shelf. This is a serious problem for our region and its ocean ecosystems. So how do we study ocean acidification? We do this, I use a variety of different approaches. We do work in the laboratory settings with aquaria. We have mesocosm experiments on large scale and in the oceans. And we also do work directly in the oceans with experiments that are done over coral reef systems. Our work at the mesocosm level in the large-scale setting has shown a direct correspondence between the capability of the coral reefs to calcify and form their skeletons and the decreasing carbonate ion concentration from left to right here. On the top, I've laid out the concentration of CO2 in the atmosphere that would be representative of these carbonate ion concentration. And you can see by a level of 560 microatmosphere in the atmosphere, the carbonate species would no longer be able to produce their skeletons. This has been reproduced many, many times, and for all coral reef systems, we find the same result. Now, a recent paper that just came out this past month have shown that indeed in the Great Barrier Reef region, over 328 Pteropods samples were studied, and what they found is a remarkable decline in calcification since 1990, as much as 14%, and an accelerating decline in the last 5 to 6 years. So there is an indication now from field observations that what we are seeing in the laboratory has some validity. Another way of looking at these kinds of experiments in the field is the work that is being done in Bergen with mesocosm experiments. These are large bags that are put out into the water column, and they have a controlled CO2 level. These are done at the 190 parts per million, which is the pre-industrial CO2 level, 370 parts per million, which is the present-day level, and 700 parts per million, which would be the level we might expect to see towards the end of the century. And we've looked at coccolithophorids, which are calcifying plants, foraminifera, and pteropods. The foraminifera and pteropods are primary food source for our fish and other mammals and birds. And so we are seeing then that the changes that are taking place with these organisms through the food webs, the pteropods in particular, which are on the bottom, they are the primary food source for the juvenile salmon and pollock and other species that we depend on for food. And what we find in these experiments is a very striking decrease in calcification rate, anywhere from 9 to 45%, and a deformation of these calcifying organisms. Indeed, some of the organisms actually no longer calcify at all in the high CO2 concentrations. With respect to foraminifera, we find that both in the laboratory and in the field, that there is a decrease in shell weight, a thinning of the shells, and we can see this in cores from recent sediments as well. We find about a 14% decline in the shell weight with concentrations as high as 780 parts per million. Now, with the pteropods that I mentioned earlier, my colleague Vicky Fabry from the Scripps Institution of Oceanography has shown to us that the living pteropods, when placed in the high CO2 waters, begin to have their shells dissolve while they're still alive. And you can see here the dissolution of their shells. And this means that they will no longer, in some places, have a place where they can form their shells if we continue along this pathway that we're on. With closer to home, we look at the effects on mussels and oysters, and we see the same decreasing calcification of the mussels and oysters, anywhere from 10 to 25% with increasing CO2 levels. And indeed, if you look at the juvenile species, the clams, for example, when they're just formed the first time, within 24 hours, there's massive dissolution of the shells. That's the whitening of the shells that you can see. And within two weeks, the shells are gone and the animals have died. And you can see this processes in very short order in real time along our coast in New England. Some very exciting new work has been done by my colleague Gretchen Hoffman from the University of California, Santa Barbara with sea urchins. She shows that sea urchins show a very declining calcium carbonate structure of the larval forms of the sea urchins. And she has formed some very exciting new genomic research to show that in a higher CO2 levels, there is an expression of the genes that control calcification, and it's a downward expression, not only for calcification, but for metabolism and other processes. And it's this genomics research that we will be able to understand mechanistically what the capabilities and the limitations for calcification in the future. So this is a very exciting new area of research that's going on. When you think of just calcifying organisms themselves, such as bivalves, clams, scallops, mussels, echinoderms, we're talking about within the United States a $2 billion a year industry. In fact, the shellfish industry within the United States is half the entire fishing industry within the United States. So this is an important issue. Not only does ocean acidification affect calcification, but it also affects things like fertilization rates for organisms, their ability, the egg and sperm to come together, increased juvenile mortality, particularly in fish and fish larvae. Here's an example where fish larvae were put in high CO2 waters, and there was anywhere from 60 to 100% mortality of fish larvae. So it is the very young larval forms that are more susceptible than the adults in most cases. And finally, there's reduced growth in the adults for many species, impaired oxygen transport for squid and other organisms. Squid depend on oxygen transport for them to capture their prey, so this is particularly important to them. And there's reduced metabolism for most organisms. Now, the other issue is what happens as we have impacts at the low end of the food chain. How does that affect the high end of the food chain? And I show you this simple pathway here from coccolithophorids, which are the marine plants, to the copepods, to the fish, or from the coccolithophorids to the pteropods to the fish. And you can see what happens at the food chain will have a significant impact on the fish. And here's one model output that shows that a 10% decrease in pteropod production leads to a 20% weight loss in mature salmon weight. So there is a direct connection through the food chain that we need to be concerned about. Now, there have been studies to show that these impacts can be seen naturally in the open ocean in areas where there are significantly higher CO2 levels naturally. And this is an important study that was done in Italy where there was a natural CO2 seep from volcanic processes, and it was in a coral reef bed. In the region of the very high CO2 and low pH waters down to 6.8, we saw a loss of the coral reef species, and in fact a loss in most of the calcifying species, but an intensification and increase in the seagrasses. So you can tell from this that there will certainly be winners and losers, but how this permeates through the ecosystem, and particularly as important ecosystems like our coral reef ecosystems that provide fish resources for about 25% of all our fish resources. These are very serious issues that we need to concern ourselves. In this particular study that was done, it was shown that the calcifying organism went through a major change at a pH of about 7.8 or so. And this may well be one of the first examples of a tipping point. And I show these last two pictures here on the bottom, showing again we see where the organisms that are in these areas, their shells are dissolving while they're still alive. If you do a scorecard on what we know so far, it sort of looks like this. For all the calcifying organisms, we see almost all the organisms showing a declining response. There are one or two exceptions with the coccolithophorids that have different responses. This means we have to look at the species by species differences. For many of the other organisms, it's just a negative response. Photosynthesis, interestingly enough, there's a positive response with CO2, and this means we may be seeing a very significant shift in photosynthetic organisms from the way they are to what they might be in the future.
This is particularly true for cyanobacteria and nitrogen fixers. So our entire ecosystem might change very dramatically, and this then would provide a feedback mechanism to the atmosphere that we should be concerned about in terms of the ocean's role in global climate change. And finally, reproduction is significantly affected as well, from what we can tell right now. So to be fair, we need to explain ourselves in a clear of what we know and what we don't know. Most of the experiments thus far have been very abrupt with high CO2 and pH changes with specific species strains, with short time duration experiments and large pH changes. So there's a lot we don't know yet, particularly responses of genetically diverse populations, synergistic effects with temperature, trace metals, and other species, particularly temperature and CO2, have shown deleterious effects for coral reef organisms, species replacements, community responses, and as I said, impacts the global carbon cycle. NOAA, USGS, National Science Foundation, and the NASA have all been working together to develop a national program on ocean acidification. And we have developed approaches to develop technology to observe it in the oceans, to model this with models, to do more physiological research studies, and have interagency program on ocean acidification. I do want everybody to know that there is now the capability of measuring ocean acidification on moorings. Here's the example of the very first mooring that has been deployed in the Pacific Ocean. And here we're measuring the PCO2 and the pH on a mooring, much as we would do with temperature, salinity, and oxygen. So we have the capability to monitor these processes in the oceans in real time. So I'll leave you with this thought. This is from my friend Carl Turley. This is the sunset of the oceans with man's footprint now detectable. It's warmer, it's more acidic, it's less diverse, and it's overexploited. The oceans, for certain, that we know that it will become more acidic. The way of reducing the impact of global ocean acidification is reduction in CO2 emissions, and we need a concerted national and international effort to reduce the uncertainties, vulnerabilities on our marine systems as soon as possible. Thank you.